So in the morning session we discussed about karyotyping, right? So karyotyping. So today I will discuss about fish, and I will discuss about flow cytometry. Okay. Other one I know. CGH, comparative genomic hybridization, that we will discuss tomorrow morning. Okay? CRISPR, okay, CRISPR is easy. Fine. So listen, for your exam point of view, you will get 10 marks definitely from flow cytometry or karyotyping. I'm not sure about fish and CGH, but flow cytometry is very important topic for exam. Okay? So pay attention. So first I will start with karyotyping already finished. So simple. First we are taking the cells. Okay. When we are culturing them, culturing will allow the cells to divide. When they are dividing, we are arresting them in the metaphase. We take colchicine, right? After that we are going for staining. Okay. So I didn't say this previously. There are two types of stains using for chromosomal staining. Okay. G banding, G staining and S staining. G banding or S banding. Okay. G staining, S staining. Now simply students, when we are staining a chromosome, they will show light bands and dark bands. Okay, light bands and dark bands, and they have a proper sequence. Okay, just like a barcode, each chromosome has its own light and dark bands. Okay, so that banding also we need to do. That is optional. If you are going for banding, then we can identify chromosomes for banding. See, after doing this banding, do you think the chromosomes will be properly aligned like the way we are seeing in the photos? That's not possible, okay? All chromosomes are scattered in the cell, okay? Now we are going to take a photo of the scattered chromosomes. In the cell, there will be many chromosomes like this. Randomly they will be scattered, okay? Now there is a photograph technique called karyogram, simple. We are taking a photo of a chromosomes. Now the software will automatically arrange the chromosomes. After taking a photo, the photos will be stitched in that way that it will collage all the photos. 23 pairs of chromosomes will be collaged. Okay, that concept is called as karyogram. Karyogram. Then we will get the chromosomes, that's it, 23 pairs. Okay, so this is the typical workflow of karyotyping. Now, the next one was Philadelphia chromosome, or maybe there is a mutation in a chromosome. So let us take a mutation for muscular dystrophy. Okay. We know that muscular dystrophy is caused by a mutation. Maybe mutation present in the chromosome number 6. In the chromosome number 6, we have P arm and Q arm. Just for example, I am saying. Now, I assume the region of this mutation or this gene was in this location. Okay. So now I extracted. I know that here there is a gene. Okay. I just want to know, this is a very small mutation which we can't find with a normal karyotyping. It's a small inversion of deletion, slight change in the chromosome that we cannot able to differentiate with a simple staining procedure. How to identify such minute mistakes? How to identify minute changes in the chromosome? That is the challenge. Okay, how to identify minute changes in a chromosome? Okay, for that purpose, now see, I have taken a piece of DNA, okay, and I have unwinded this portion. This portion has been unwinded like this, okay. DNA has a, okay, then complementary base pairs are there, 5' to 3', 5' to 3'. Okay. This is the gene, let us assume, and it has base pairs. Okay. I forgot the word. We are using an enzyme, this nuclease. This enzyme will cut the DNA in random places. Nuclease will cut the DNA in the random places. Clear? They will remove some base pairs. They will remove some base pairs like this. Okay? Randomly they will purposefully remove some base pairs from the gene. Okay? Now, removal of this base pairs leads to a type of DNA strand with missing base pairs. This is called as introduction of nicks and ticks. Nicks and ticks. I hope you know the meaning of nicks and ticks. Rough. The DNA becomes rough. There are no sequential base pairs. Okay. There are gaps. Then, now we are taking base pairs. Adenosine, tyrosine, guanosine, uracil, or cytosine. Uracil in case of RNA. Okay. Now what I am going to do is, to this base pair, I will add a fluorophore. I took adenosine and I add a fluorophore. Maybe something iodine fluorophore, green color. Then to the T, I add a fluorophore, cyanide, pink color. This is another blue color, yellow color. Simply, I radio tagged. I tagged base pair with a fluorophore. What is the significance of fluorophore? What is the significance? Under UV light, if we hit a UV light on a fluorophore molecule, it will glow very brightly. Very brightly it will glow under microscope. Clear? I know this is last lecture, you are tired. So that's why I like morning lectures. You will be active at that time. Listen then, in a nutshell, simple. I removed the base pair students. Okay. DNA has one good characteristic. The good thing was it will self-repair. We already stated. Whenever there are gaps, proofread, it will do the proofreading and it will self-repair the gaps. Proofreading, simply. So I removed the base pairs. Then I took another suspension with base pairs with radio tags. Then what I will do? I will add this radio tags in this pool, in this solution. Now, whenever it is trying to repair a T portion, now it might take our radio tag with T. It will take our radio tag with T to repair the DNA. Got it? So this particular portion of it, only this piece of RNA I took. That is the primer, simple primer. Clear? Now, suspected gene. I know my suspected gene, that is muscular dystrophy. I know that. See, this is our radioactive base pair. Synthetic primer. Synthetic primer. A normal primer. Synthetic normal primer. In the normal person, they should have A, T, T or G. A, G, T, G. For example. And I radio tagged two things. Fluorophore tagged G. Okay. Always we know that it will bind with the complementary sequence. When it will bind, when the complementary matches with primer. Am I correct? Complementary must match with the primer. The primary strand. A should tag with the T, C, A, C. Right? If it is not matching, our primer won't bind. Got my point? If the now the complementary is the patient's DNA. I took a suspected maybe Psychiatr's DNA, assuming Psychiatry has muscular dystrophy. Okay. Then I have created a muscular dystrophy primer. The primer with proper sequence of muscular dystrophy. If this primer binds with the Psychiatry DNA who has muscular dystrophy, what happens? It will bind and I can able to see a glow in this chromosome in one particular region under UV microscope. Simple. If you don't have mutation, I won't get any binding there. The chromosome won't glow under UV microscope. That's it. This is fish. This is called as hybridization. Hybridizing a primer to the pre-existing DNA with the help of fluorophore tagged nucleoside primer. With the help of fluorophore tagged primer, we are hybridizing this synthetic primer with the DNA to check whether they will bind or not. Binding is for hybridization, no binding, no hybridization. And all these things happening in a living cell, not outside the cell. Hence it is called as fluorescent in situ, outside the cell. We destroyed the chromosomes. In situ means outside, in vivo means inside. Fluorescent in situ outside the cell, hybridization. Hybridization means binding again, annually, simply. Okay? This is called as fish. I thought of discussing flow cytometry, but by seeing your faces, I'm not interested now. Tomorrow morning we'll discuss flow cytometry. Let us see a strong animation of fish now. Then you will get some clarity how fish works. So in a nutshell, this is the idea, and let us see what this YouTube was saying for animation. Is it on? So direct I'm going to the topic. I think I need to mute it, it's not very copyright. Yeah. Yes, see here students. So this is the fish fluorescent in situ hybridization. How to stop it? Yes. Actually I want the audio. I will do one thing. Let me pause this video.
Oh, I don't have pause option. Leave it. I have another